The Black Queen hypothesis is a theory of coevolution. It derives its name from the more popular theory of coevolution, Red Queen hypothesis, which indicates that a species constantly needs to evolve in order to survive. The Black Queen hypothesis is a theory of reductive evolution that suggests natural selection can drive organisms to reduce their genome size. In other words, a gene that confers a vital biological function can become dispensable for an individual organism if its community members express that gene in a leaky fashion. Reductive evolution is the process by which microorganisms remove genes from their genome. It can occur when bacteria, found in a free-living state, enter a restrictive state, or are completely absorbed by another organism, becoming intracellular symbiogenesis. The bacteria will adapt to survive and thrive in the restrictive state by altering and reducing its genome to get rid of the newly redundant pathways that are provided by the host. In an endosymbiont or symbiogenesis relationship, where both the guest and host benefit, the host can also undergo reductive evolution to eliminate pathways that are more efficiently provided for by the guest. The Black Queen hypothesis posits that some genes and their biological functions are very costly to maintain, and thus dispensing of them provides an evolutionary advantage to the individual. However, these functions or genes are extremely important for the survival of the community as a whole, causing it to be retained by certain members, leading to commensalistic and or mutualistic interactions. Compared to the Red Queen hypothesis, it is a fairly recent hypothesis, thus, it has not been thoroughly tested, and the mechanisms driving it have not been fully elucidated. Although, currently Black Queen hypothesis has been used only in the context of microbial communities, the hypothesis is gaining traction as more research continues to be conducted. In Black Queen hypothesis, shooting the moon refers to the strategy in which a helper for one function is more likely to become a helper for another unrelated function. As a result, such helper organism will retain all genes encoding leaky functions, carrying and maintaining a large genome, which might seem maladaptive. However, during events leading to a population bottleneck, this strategy can lead to greater probability of survival of these leaky genes, essential functions, in the community. When an organism loses all its leaky functions, it can grow faster, by virtue of a smaller genome and faster replication times. Therefore, in a population bottleneck, organisms will need to cheat on each other for all required leaky functions, leading to the evolution of mutual dependencies. An example is the dependence of marine bacteria on helper organisms to protect themselves from hydrogen peroxide. Later, it was extended to explain nitrogen fixation, nutrient acquisition, and biofilm production in microbes.